Hi guys, my name is Allison,
I'm Lavernie.
and today we'll be sharing some of our lab's work in synthesizing and investigating the biological activity of these simple yet potent arylsulfonamides, specifically through their Wnt1 modulation. So first, we'll begin with some biological background into the Wnt beta-catenin cell signaling pathway and how there have been previous campaigns into designing small molecule inhibitors that act through this pathway and how that ultimately inspired two different structure-activity relationship studies from our lab, as well as the conclusions we have reached from those studies and future directions. So to start, the Wnt signaling pathways are a family of cell signaling pathways that split into two different categories, the first being the canonical and the second being the non-canonical. And the main difference is that the canonical mainly revolves around this protein called beta-catenin. And when Wnt ligands like Wnt1, Wnt2, Wnt2b, the list goes on, bind to cell surface receptors and co-receptors like frizzled and LRP, that activates a protein called disheveled, which ultimately pulls another protein called axin away from the destruction complex. And this destruction complex is a family of protein and kinases that effectively work together to phosphorylate and ultimately ubiquitinate and degrade beta-catenin, preventing it from being translocated into the cell, activating transcription factors TCF and LEF, which ultimately upregulate genes like c-Myc and cyclin D1, which ultimately, when overexpressed, will cause cancer. And specifically relating to cancers that are derived from mutations in this pathway, most of them cause colorectal cancer. There are a few others like ovarian cancer and hepatocellular cancer. But most of the time, these cancers are caused by mutations more downstream in the pathway, as you can see in the pie chart, rather than, for example, mutations in the actual Wnt ligands themselves. And usually this occurs in the destruction complex, like mutations with Axin or APC. So previously, there have been FDA-approved drugs that work through either activating or downregulating the Wnt pathway. For example, niclosamide is an antiparasitic that, if you remember LRP, which is one of the co-receptors, niclosamide basically prevents Wnt from binding to LRP6 and thus effectively inhibits the pathway. On the other hand, riluzole, which is a drug that targets amyotrophic lateral sclerosis or ALS, actually activates the pathway, and because in these neurodegenerative diseases, the Wnt pathway is actually downregulated. But for both of these drugs, a commonality is that they target things other than the cancers that are caused by mutations in this pathway. And so during that time, there was actually kind of an unexplored space in resolving drug inhibitors that could target such colorectal cancers. And so different researchers put their minds together to come up with a drug that could do so. And oftentimes in medicinal chemistry, this occurs through something called high throughput screening. In some cases, this involves doing something like molecular docking, in which you know your target of your drug and you screen through a bunch of compounds to see which has the best binding affinity to that known drug pocket. On the other hand, for Huang and coworkers in 2016, they conducted something called a luciferase assay, which I'll explain more in detail later, to find an optimized drug that could act selectively in the Wnt pathway. Essentially, through measuring luminescence, once again, I'll explain the mechanism of this assay in a few slides. They identified compound MSAB for short, which had an IC50 of around 0.583 micromolar. And usually for a hit compound, usually the IC50 should be in the nanomolar range. However, the remarkable thing about this compound is that despite being such a simple arylsulfonamide that can be synthesized in one step, it's highly selective towards Wnt-dependent cell lines and has decent potency as well. And they did try conducting some studies into investigating what this compound could potentially be targeting, and they came to the conclusion that MSAB would bind to the armadillo repeat section of beta-catenin. And so proteins have an N-terminus and C-terminus, and for beta-catenin, the armadillo repeat is the middle section of amino acids in that protein. And the way they determined this was through a variety of NMR experiments, as well as Western blotting with truncated versions of the protein to see where it would best bind to. However, this hypothesis was kind of challenged in 2022 by McCoy and coworkers, who found that through their NMR experiments with this compound, the results showed that the binding affinity suggested via NMR studies was not enough to explain the high selectivity towards Wnt-dependent cell lines and good activity of this compound. So as of right now, where MSAB binds to in biological systems is still ambiguous. However, it's still evident that MSAB is a potent Wnt1 modulator. And so that led to our first initial study, structure-activity relationship study, which involved modifying the ester position and length on the right-hand aryl ring of MSAB. And so with this in mind, we synthesized 3 analogs of MSAB, first changing the position of this methyl ester in red from the 3 position or meta position to the 4 or para position as this compound M4, and as well as 2 other analogs with ethyl esters instead of methyl esters at the 3 and 4 positions respectively. And what we found was through cell viability assays that MSAB still performed the best. So essentially changing the length and position of the ester on that aryl fragment would decrease the potency. And to summarize the data, we have a table of IC50s, which are essentially like the concentration, the lowest concentration of drug required to kill half the cells. So we tested them across 5 different Wnt-dependent cell lines. We have colorectal, breast cancer, and human embryonic kidney cancers, or human embryonic kidney cells. And you can see that the top row, which is M3 or MSAB, has the lowest values, meaning that there is the least amount of drug required to kill half these cells. Additionally, to evaluate these compounds and make sure that they actually acted through the Wnt pathway, even though the actual biological mechanism is not known, we want to confirm that they acted through the same pathway as MSAB. We performed a luciferase assay, and this luciferase assay I mentioned earlier when I talked about Huang and coworkers finding MSAB through high-throughput screening with this assay. And essentially how it works is instead of drugging regular cancer cells, we drug these engineered cells or reporter cells that are engineered to encode a certain gene called luciferase. And when luciferase reacts with luciferin, which is in the luciferase buffer solution that is used to measure the luminescence, it produces light, and that's a measurable quantity that can be used to then determine how well these compounds perform. And in order to express luciferase, we add a compound called CHR-99021, which is a selective GSK3 beta inhibitor. And if you recall, GSK3 beta is part of the destruction complex. And when you inhibit this protein, or this kinase, it effectively causes the destruction complex to not be able to function. And that means beta-catenin is not allowed to be ubiquitinated and proteasomally degraded. And so this means that beta-catenin is overexpressed in these cells. And if the compound is an inhibitor in the pathway or more downstream of the destruction complex, in the destruction complex, that means that you would see a lower luminescence activity because more beta-catenin is inhibited, meaning there's less luciferase that is expressed. So what we found was that all of our compounds were indeed acting through inhibiting this pathway. However, similar to the cell viability data, methyl-3 or MSAB still performed the best with and without CHR. And the better activity seen with CHR-99021 compared to without CHR is due to the fact that it's overexpressing beta-catenin. And so unlike the regular cells where it's not as, the levels of beta-catenin are not as high, you would see better activity with your drug.
So moving on to the second portion of our SAR study, we started off with an optimization and condition screening of various reported conditions, as well as using Lewis acids to further accelerate the reaction. Since in the previous 1.0 study, our our reactions were really suboptimal with really long conversion times as well as really suboptimal yield. So we aim to change that before making all of our analogs. So we discovered that pyridine in THF as the solvent gave us the best conversion time as well as good yield. And it produced spot-to-spot conversion for almost all of our sulfonamide analogs, and the reaction times were relatively really short, with most of them finishing at around like 7 hours maximum, with some exceptions, of course. And additionally, we developed a workup method that effectively removes all the remaining starting material, such as methyl 3-amino benzoate. And additionally, excess pyridine is removed through copper sulfate since it is a Lewis base. And so here's kind of like the reaction mechanism. Oh, wait, can you go back one slide? So essentially the lone pairs on the amine attacks the sulfur atom on the sulfonyl chloride. And then that essentially forms like a tetrahedral intermediate, which is relatively unstable. So it kicks off the chlorine, forming HCl and your sulfonamide product. So then we began to synthesize our analogs, which involved various monosubstituted analogs, such as our para-substituted, meta, and ortho-substituted analogs, as well as different multi-substituted rings, for example, like the 2,5-chloral, which is like different positions on the arrow ring. And we also have some of our other analogs, notably like the danzol and biphenyl, as well as like our propyl analogs. So it's kind of just to like get a good idea or a good grasp to see if we can observe any structure-activity relationship. And lastly, we decided to synthesize these amides because they have very similar 3D geometry to sulfonamide, and it would greatly increase the scope of our project as some of these starting materials are much more accessible than the sulfonyl chlorides. So when the R group here is a chlorine atom, essentially it goes through like a nucleophilic substitution reaction, and when the R group here is an alcohol, we do an amide coupling to essentially form this amide here in the middle. So moving on, we tested our analogs in a series of cell viability assays involving using various Wnt-dependent cancer cell lines. For example, SCOF3 is an ovarian cancer cell line, and HT29 as well as CT26 are colorectal cancer cell lines. And essentially how a cell viability assay work is after you drug your compounds in your cells, you add a reagent that is yellow called MTT, and essentially living cells metabolize this yellow compound into a purple compound named formazan. So by quantifying how purple your wells are, essentially you're able to determine the cell viability of your compounds in these specific cancer cell lines. So here's kind of like the notable results that we got from this series of cell viability assays. So what we noticed is the 4 substituted analogs, such as like the 4-iodo, had very comparable potencies with MSAB, and it was also noted some of the other ones like danzol and benzoloxy also showed pretty good potency, and this is kind of like a trend that we observed throughout various different cancer cell lines and not only in one specific cell line. So kind of the trend that we can get here is the para-substituted analogs do exceptionally well in cell viability assays. So MSAB itself is a kind of like a para-substituted. It has a methyl group at the para position. And our lead compounds, including the 4-iodo, 4-chloral, and like the other ones have halogen substituted ones. And then we also have 4-nitro and 4-benzoloxy being pretty potent. And so what we can really deduce from here is that by changing the fourth position to, for example, the meta or ortho position is not well tolerated in kind of like the biological activity of these compounds. And so MSAB itself is a single micromolar, has a single micromolar IC50 values amongst different cell lines. And so we can see here some of our compounds here in blue show very comparable potencies. And you might be wondering why the hex cells in the middle here don't demonstrate the same potency even with MSAB. It's because they're human embryonic kidney cells and not cancer cells. So it's actually a good thing that we see no exceptional killing with our analogs in this cell line. So, building on what Allison said earlier, we decided to run a firefly luciferase assay on our analogs. And so what we see here is that our compounds do decently well at high concentrations in terms of inhibiting the luminescent signal, which is signify kind of like inhibiting the Wnt overactivation activity. And so what is really interesting, especially with these compounds highlighted here in red, is that, for example, the 3-methyl and 3-chloro in the middle showed really good potency, but it did not reflect the same kind of like same kind of potency in cell viability assays, which would lead us to kind of like think that interferes with the assay itself, rather than being an actual like potent inhibitor. So then moving on, we finished off with also doing the biological evaluation of our amide analogs. So what is really important to note here is that at high concentrations, these amides actually show kind of like killing of the cells, so kind of moderate killing of the cells, so not very potent, but also it demonstrated some partial killing. And next we ran the same firefly luciferase assay on these amides. And what we saw here was something pretty exceptionally rare, with our compounds showing almost 100 times the signal compared to our positive control when we overexpressed beta-catenin with CHR. And essentially, that is a very interesting trend, since such simple molecules, such as these bioaryl-like compounds, usually aren't very selective, and essentially it shouldn't be able to be such a potent Wnt agonist. And then later we also ran the firefly luciferase assay at the same time point, 24 hours, without CHR. And what we also saw was around like a 1.5 to 2 times increase in Wnt activity, which is consistent with the results, since you don't expect to see a lot of beta-catenin in the cells since you're not overexpressing with CHR. And so this would kind of lead us to believe that these compounds may be inhibiting like the degradation of beta-catenin out of the cell, which would lead it to become trapped in the cell, and ultimately we will get a higher signal. So more recently, we did a co-delivery assay with CHR. So instead of doing luminescence as we previously done, we quantified cell viabilities using the reporter cell line, the 3T3 cells we used for the luciferase assay. And while you may think that some of these results are not the most optimal, it is important to note when you overexpress CHR at the quantity that we're using, for example, 10 micromolar, we essentially inhibit the entire destruction complex of such as GSK3 beta, since the IC50 value of CHR is around in the nanomolar range. So we would expect to see a lot of cell proliferation, which our compounds at the highest concentration are actually still able to kind of inhibit the activity, so lowering the cell viability. And so we also see without CHR that our compounds also do some sort of like killing of the cells, but it is like, so we didn't really come to very conclusive results with this assay, and something we want to replicate in the future. Lastly, we decided to, as a true medchem effort, we decided to dock these molecules at the reported binding site of MSAB, which is kind of this place in the armadillo domain repeat where it interacts with the TCF and LEF downstream, essentially like receptors. And what we really got was like suboptimal docking scores, which would indicate not very potent binding to this specific binding site. But what was the interesting trend that we observed from our biological assays was that as your halogen groups get bigger, you actually see this sigma hole formation. For example, in your iodine, you see this like dark blue spot, and that would be a sigma hole, which is essentially like an electron deficient region, which is able to actually form like various interactions with the carbonyl of different amino acid residues, which would further prompt us to believe that some of our best analogs are really potent due to some halogen bond effect. So our future directions would to run NTT assay and luciferase assay on some of our newly synthesized analogs. And of course, we would love to replicate assays on existing compounds in order to get better curves, cell viability curves, etc. And additionally, we want to investigate the amide activity since it is really interesting how switching from a sulfonamide scaffold to an amide scaffold would greatly change like the biological mechanism of these compounds. And of course, we'd also love to determine essentially where MSAB binds to and essentially probe the mechanism of inhibition in cells. So thank you to our other group members, as well as our advisor, Dr. Edward New, who has provided tremendous guidance in our project, as well as ASDRP for providing the resources for our project. And additionally, we have the preprint out from our first initial study. So if you're interested, the QR code is up here and feel free to scan it and take a look. Yeah, thank you.